This is the BioCentury Show. Brought to you by BioCentury Grand Rounds. Join BioCentury and Insights partner McKinsey & Company this September in Nashville for an R&D forum at the interface of industry and academia.
Hello and welcome to a special episode of the BioCentury Show. I am Simone Fishburn, Editor-in-Chief at BioCentury, and it is a pleasure to welcome Vasant Narasimhan, CEO of Novartis, to discuss the data Novartis is unveiling this week at ASCO and at the Allergy and Clinical Immunology Conference, EAACI. Vas, welcome to the show. Now, I'm not going to go into your background. I think our audience is well familiar with you and with Novartis, and I will refer them to our previous conversations at biocentury.com, where you and I have discussed and you've laid out the plans for the company. We'll get into some of that strategy, but I want to start with the oncology data for your phase 3 compound, Scemblix. Now, Scemblix is a next-generation tyrosine kinase inhibitor for CML, and that, of course, is an area that Novartis forged ground. I looked it up. It's 24 years ago to the month,
That's right.
I think, that Gleevec was approved, right? I would look at this, in fact, as a third generation of that class, with a compound in Scemblix that acts at a different site on BCR-ABL. We'll talk a little bit about that maybe. But I want to start with the data. Your goal is to move this to the front line in CML. So to what extent do you think the data support that?
Yeah, great to be with you, Simone. Look, I think this is landmark data for the CML community. What we know is that in CML, despite the advances of Gleevec and second-generation TKIs, there's significant unmet need. We know 50% of patients don't reach goal in their first year. 25% of patients have significant issues staying at their recommended dose level, and hence you see patients moving to second-line, third-line therapies. In this study, we put Scemblix in the front line versus Gleevec or an investigator choice of TKI, and we showed a pretty impressive result when you look at the superiority we showed in the overall study, and as well in Gleevec alone. And so I think on the efficacy side, we clearly demonstrated a significant improvement on major molecular response. But then when you go deeper into the data, there's also significant improvements in deep molecular response, which really matters to CML experts. So really hit on the efficacy side of the equation. But what I think is even more impressive in many ways is on the safety side. So when you look at this, we had significantly fewer SAEs and also significantly fewer AEs leading to dose reductions. So patients were able to stay on their target dose of the medicine, which we believe will enable more patients to get to goal in the frontline setting. So the study really hit all of the benchmarks. And I think what's really interesting about this whole story is that here we found a different pocket on the BCR-ABL molecule. And this pocket is what enabled us to really, I think, make the next breakthrough. What it allows this medicine to do is be much more specific for this particular tyrosine kinase without hitting all of the other tyrosine kinases that the other TKIs hit.
So, right, I'm going to unpack some of that and come back to some of the points you raised. I want to start with something. You constructed the trial, as you said, against investigator choice, and you had a subgroup that is Gleevec-imatinib. Do the data sort of suggest that the biggest improvement is over imatinib rather than the second generation TKIs, in terms of the efficacy at least? And if so, how important is that distinction for moving to the first line?
Yeah, so we were superior to Gleevec, as you mentioned, and that difference was 69% versus 40%. So nearly a 30% difference in major molecular response. We weren't powered against the second-gen TKIs. It was not something the regulators asked us to do, but we were numerically superior in the secondary endpoint as well to the second-gen TKIs on the order of, my recollection is 8 to 10% on major molecular response. So we were numerically superior there. So I think we demonstrated we meet the efficacy bar. Now, I think what most clinicians would say, the second-gen TKIs are much more onerous from an adverse event profile. So that's where the safety comes in. So you have a much more high efficacy versus Gleevec, a safer medicine versus the second-gen TKIs. And I think that positions us well to be the very hopefully preferred first-line agent.
So one of my questions and thoughts about this is, in these 24 years, CML has really transformed as a disease. At the same time, patients really take the medications for life. So what are the chances that with this strategy, patients could someday come off treatment? Is that on the radar?
It is always on the radar, and historically with Tasigna, we ran treatment-free remission studies to try to get patients to that goal. And there are a subset of patients we hope also with this medicine will be able to get there. The reality is most patients still progress. What we don't know yet with Scemblix, if we were to give it in the frontline setting, could we, of course, reduce the number of patients? It would get more patients to that goal and then maybe roll patients off. That's going to be a longer-term, I think, story. That will take 5 to 10 years plus to generate that data. In the meantime, our goal with Scemblix is to position it frontline. We have ongoing studies as well in combination with Gleevec, as well as in the second line, and it's already approved in the third line. So really, it becomes an agent that could be used in any line of CML.
So, you got straight to my heart before because I'm a molecular pharmacologist. I want to talk about the molecular strategy.
Yeah.
This molecule binds a different site, the myristoyl binding site of BCR-ABL, rather than the ATP binding site, which is obviously where you get your differentiation from other TKIs. Now, it also makes sense that that can be effective in resistant cancers. What I'm wondering is whether you have a molecular hypothesis or if there's any reason to believe that that mechanism might underlie differences in the adverse event profile compared with other tyrosine kinase inhibitors.
Yeah, we do. I mean, I think first on the efficacy side, we think that pocket is more stable to hit. And so we think that that might be part of the reason why we see the deeper molecular response from an efficacy standpoint.
And it's having an allosteric effect, is it?
That's right. So
right.
In one of the allosteric positions, we actually have a very high affinity to that pocket. Now, what's interesting is when you look at the TKI tree of kind of where are the off-target hits, certainly Gleevec has a certain number of off-target hits. The 2 second-gen or 3 second-gen approved TKIs was 3 or 4, depending on how you count. They have quite a wide range of off-target TKIs hits that cause the safety issues. Now, with the case of our medicine, Scemblix, and hitting this myristoyl pocket, it's very, very specific. We see the tree almost silent. And so it looks like with this medicine and hitting this pocket, there isn't a lot of similar pockets on other TKIs, and hence we get the high specificity and the better safety profile.
Let's stay with innovation. The endpoints are really interesting. Major molecular response, MMR, is the primary endpoint. I think just the audience to point out that's BCR-ABL detection at under 1 in 1,000 compared with untreated CML, which is a 3-log reduction. You referred to deep outcomes, MR4, MR5, which are 4 and 5-log reductions. So as efficacy gets better in this field, how are you thinking about the relative importance of deeper outcomes versus durability, say, over 96 weeks? What do physicians care most about?
It's interesting. We are very interested in deep molecular response because we think that deep molecular response is what might lead to a functional cure, right? Or something where actually, if you get a deep molecular response that's sustained over time, patients can come off drug. I think for practicing clinicians, it's really that 96-week period to really get comfortable that the efficacy is sustained. And we certainly see that so far with Scemblix. I think for the long run, in terms of making sure that these patients can stay at a controlled state of disease, that deep molecular response gives me confidence that we should do better over time than the other agents. And you see that in the data sets, pretty significant improvements in deep molecular response. I think an open question that we often get and we still need to investigate is if you hit both pockets. So if Gleevec is hitting the ATP pocket and if Scemblix is hitting the myristoyl pocket, is that going to give you an even deeper response? And that's been something we've been interested in and certainly something we're going to study going forward.
And, we're seeing in other fields, I think multiple myeloma, others, we really are seeing evolution of different endpoints in order to show differentiation. Is that something that you're also focused on here? I mean, we've talked about deep responses, but are there different measures of endpoints?
I mean, to date, regulatory agencies are really focused on major molecular response. I think what the issue with deep molecular response is because some of the variabilities in the assay, there's not enough comfort yet to use that as a regulatory endpoint. We certainly tried to use deep molecular response as another endpoint in this study from a regulatory standpoint, but ultimately was not accepted. So I think for now, we're still in the major molecular response world, at least for the coming years.
And that is a great segue to my next question, because CML patients have been major beneficiaries of the accelerated approval pathway, which is actually from the evolution of those endpoints from the early days to now, of course. And how are is that pathway really fundamental to the strategy for getting approval in early lines of CML? There's obviously been and I'm sure you've seen my colleague Steve Austin written about this, a lot of words, spilt on the number of patient life years, I suppose. Talk a little bit about accelerated approval here.
Yeah, I think with CML, there's enough confidence that major molecular response correlates with long-term survival. I mean, that has been, I think, relatively well studied. So I think regulators are comfortable with full approvals with hitting major molecular response goals. That's certainly what we've seen more recently in the field. Now, I think more broadly, of course, there is an open question as to how the shifting landscapes that we see. There's obviously a lot more focus now on overall survival in general from the agency, certainly at least with FDA. I think there's more reluctance to offer accelerated approval pathways unless there's a clear endpoint that can correlate with a hard outcome. And I think that is something that we can talk about maybe in another broadcast, kind of the shifting regulatory landscape, which will affect, I think, drug development more broadly over the coming years.
Well, we certainly will take you up on that and talk more about it. For the moment, we're going to take a brief break. And when we come back, I want to talk about the data on Remibrutinib at EAACI and a few strategy questions beyond that. Thanks.
This September in Nashville, BioCentury launches Grand Rounds, a new interdisciplinary R&D forum at the interface of industry and academia. Join BioCentury and a rich roster of innovators and key thought leaders to identify leading-edge discoveries and discuss urgent challenges that must be solved to translate these innovations into product development and medical practice. Meet academic innovators, physician scientists, early-stage investors, and biopharma R&D leaders for 2 days of networking, partnering, and debate. Register at biocenturygrandrounds.com.
We are back at the special episode of the BioCentury Show with Vasant Narasimhan, CEO of Novartis. And we're going to talk now about Remibrutinib, Novartis' small molecule BTK inhibitor, where you are presenting phase 3 data in chronic spontaneous urticaria, CSU. I want to start actually with your strategy before we talk about the data, because Remi really has a pipeline and a product potential. Now, we're seeing not just a massive increase in immunity and inflammation broadly, I&I, but actually specifically in dermatology. So what was your rationale for picking urticaria for the launch indication of this product?
Yeah, look, with Remibrutinib, we think it's a pretty unique profile. So here you have a potent BTK inhibitor that at a relatively low dose seems to give very strong efficacy signals and a clean safety profile. I think we first actually looked at Remibrutinib in oncology indications, but we didn't see significant differentiation from the existing BTK inhibitors, but we did observe this very clean safety profile. In particular, we think relative to even other BTK inhibitors being studied in immunology, the structure of the molecule is different, which we believe leads to fewer off-target toxicities and the clean safety profile. So then we began to think, why not move it into immunology, and particularly dermatology? And one area where we know that B-cell activation has a significant role, particularly, and we saw this as a molecule that can penetrate deep into the skin. We thought CSU could be an interesting test case. Now, I think we ourselves were surprised to see almost biologic-like efficacy with Remibrutinib as an oral twice-a-day agent. And so we took it forward in CSU and alongside a number of other indications, which we can get to. But we really think about Remibrutinib as a core immunology drug today.
And are you thinking about it as a sort of pipeline and a product? Are I know you've got 1 or 2 other indications there, but it's actually, I think, 10 years since a major new immunology drug from Novartis, and it is one of your core areas. So how big a part of the picture is this molecule?
Yeah, we're very excited about it. We think it can be already in CSU. We've demonstrated efficacy. We're going to move it as well into CIndU, which is a related indication. We have it in hidradenitis suppurativa, where we recently launched with Cosentyx. So this would give us an oral option to bring alongside that Cosentyx. And then we're also looking at it in a number of other immunology indications currently in phase 2b clinical trials. So we're trying to go broad quickly with Remibrutinib. We have a few other agents coming alongside behind it, ianalumab and a few others, but I think Remibrutinib right now is our biggest near-term immunology asset.
All right, I want to come back to some of those questions, but I do want to talk about some, the sciencey stuff. So, when we look at this indication, you reported that more than half of patients in the trial achieved USA7 of 0, right? Weekly score of 0. Now, in your past phase 3 for Xolair, in that indication, you were looking at patients who had sort of less than or equal to 6. So our question is, is the bar, did you set out with a higher bar, when you went in here, and is that bar going to continue to rise?
Well, I think from an endpoint standpoint, the endpoints have evolved. The UAS7 involves one other domain in terms of its overall, evaluation of the drug. It is a slightly higher bar, but we would say the efficacy is in line with Xolair from how we look at the efficacy here. What we think is really important about the Remibrutinib data is two things. One is the speed of onset. So you have here onset within 1 week. Most of the biologics can take up to 3 or 4 weeks or even longer, the currently approved agents. And so when you think about a patient who has hives, who's having severe itch, you want rapid relief. And that's what this oral agent seems to give. And the second thing is actually just the itch domain. So when you actually looked at the itch domain data specifically, both at the initial endpoint, 24 weeks, and then moving all the way out now to the 52-week data, we see very good control of itch. So what we think here is having an oral drug with over 400,000 patients in the US alone who have hives. And of course, if you look across the major markets, we're looking at 1.5 million patients. There's a lot of patients in need of a simple oral agent, can be used on top of antihistamines to control that itch and give rapid 1-week relief.
So again, great segue. Based on your mechanism, it sort of seems the idea of BTK inhibition is to prevent the downstream release of histamine. The trial, of course, was done in combination with H1 receptor antagonists, so the second generation antihistamines. And I think you refer this to as two-pronged attack. I would call it 2 levels, but okay, 2-pronged. So, actually, does that mean that monotherapy is not on the radar? Is it always going to be a combo here? And what does that mean for sort of the side effect profiles of the antihistamines or so on?
Yeah, I mean, in this study so far, we've studied only on top of antihistamines. I think there could be a question of could you taper down the antihistamines or remove them completely? We don't have that data yet. So that's obviously something we'd want to look into in the future. Obviously preferable to have fewer agents on board, as well as, the antihistamines have their own side effects, the sedative effects, the dry mouth, et cetera. So if we could, and actually Remibrutinib is very clean overall. So we'd love to get to that point, but we'd have to do more studies over time.
So, yeah, that's kind of helpful. So I do want to talk about adverse events around the BTKs, though, because I wasn't sure what the effect is on transaminase elevation, and we know that liver tox has been a problem for some of the competitor BTKs in the MS studies, at least. Can you talk about do you differentiate that? Is that going to be a concurrent problem?
We firmly believe with the data we have today, we don't see this signal. I mean, we see overall liver safety in line with the control groups in our studies. And we've seen that pretty consistently, both in our phase 2 programs, as well as now in our CSU programs, in our 2 multiple sclerosis trials today. When some of our competitors were placed on partial or full clinical hold, we, of course, had to submit all of our data as well to FDA.
That's in MS. So you're talking about in MS, right?
In MS. And we were not placed on any holds or any restrictions because our data sets are clean. Our hypothesis, based on what we see, is, again, when you look at the structures of these molecules, our structure looks fundamentally different than those other BTK inhibitors. We posit that we can prove that this is off-target toxicities that you're seeing on the liver, on hepatocytes, that we in our in vitro experiments don't see with respect to remibrutinib. So we feel pretty good now about the safety profile. I think we'll have to see. It is notable that in the MS studies, you are using 4x the dose than what we use in our immunology studies.
Right.
So we'll have to watch in MS in particular very carefully. We think in the immunology studies, whether it's HS, whether it's CIndU, whether it's food allergy, other indications we'll go to, at this 25 milligram BID dose, we feel pretty good with the safety profile.
And, you talked about safety, but with that, you're talking tolerability as well, because, I mean, I would assume that if you don't have at least as good tolerability, your path to uptake is going to be hard.
Yeah, tolerability so far looked very, very good. I mean, we don't see dropouts in these studies. We're very well aware. I mean, our goal here is going to be positioned ahead of Xolair and Dupixent. I mean, in the treatment paradigm, we want to be the oral agent of choice that's used after a patient is uncontrolled on antihistamines. You add on remibrutinib ahead of biologics, and we see that as a significant opportunity because I think for many allergists, dermatologists in the US, about 85% of these patients are treated by allergists, 15% by dermatologists. It's preferable, we believe, to use an oral agent before having to go to biologic treatments for the long term.
So, as we've analyzed this field and we look at atopic dermatitis, I mean, that opportunity has just massively grown. I've talked, of course, to Paul Hudson from Sanofi about that. As you can imagine, he's quite excited about that. And
yes.
So, my question is whether urticaria has the potential to become the next atopic dermatitis. What are the parallels or limitations of that parallel?
Look, there's certainly the possibility that urticaria is significantly underdiagnosed. We've been discussing that as a team. How many patients are not treated simply because they don't want to go on a biologic or undertreated? There could be a much larger market opportunity than what we currently estimate in this 1.5 million to 2 million patients. Certainly on our minds. First job, of course, will be to get the drug approved and launched, but we think it could be a very significant opportunity over time. And I would note that we believe that, again, with the high efficacy we have and the rapid onset, we think we have, in this case, a better profile than the atopic dermatitis drugs, at least in CSU.
So I want to just end this part on talking about MS, multiple sclerosis. So the CD20 mAbs are really running away with that market. Ocrevus from Roche, Novartis' Kesimpta. And, it's hard when you look at the sales today to imagine where this is going to go. But do you actually think that this mechanism, BTK inhibition, can compete and disrupt that market? How big a share are you looking for there?
I think a lot of it is going to come down to the data. As you know, Simone, one of the competitors in this field read out and was not superior to Aubagio in a head-to-head study. So I think there's an open question. Is BTK inhibition as efficient as CD20 in knocking down plasma B cells? And I think that is something we will have to see. CD20 molecules are extremely efficacious. We see that on all of the relevant MS endpoints, certainly importantly on relapses, but on the MRI endpoints, on disability. I mean, they're very good. So I think it's a high bar. There's always interest, as we learned with Gilenya many years ago in the Novartis experience, in bringing an oral drug into MS. And if it's a high efficacy, safe oral drug, it certainly has a lot of opportunity. But I think the jury's still out on BTK inhibition in MS.
All right. Yeah, I think we're going to be watching that as well. As you said, as I pointed out, there's been some toxicities. I think it's interesting to have new mechanisms, but we'll see where they go. I want to zoom out to some bigger picture questions and some questions that are away from these two particular conference data that you've released. So the two that we've talked about are both small molecules. You know where I'm going to go with this, right? Okay. Now, we see actually in our data, we see continued investment, even at the early stages, in small molecules. So in terms of the Inflation Reduction Act, what's going on? Is it wait and see? Is it not as big a problem as people thought? How is it factoring into decision making?
I think it's having an impact, but I think what you're seeing is that small molecule development is very focused on areas where you see less exposure to the IRA. So if you take these two as examples, Scemblix is a medicine for a rare disease. It's carved out under the orphan. It has an orphan drug designation. It's carved out from the IRA. If you think about a chronic spontaneous urticaria, it's primarily a disease of younger people. We expect Remibrutinib in its target indications to have relatively low exposure in the Medicare population. What I expect you're going to see as you parse that data over time is fewer and fewer small molecule drugs in indications that are predominantly in the Medicare population. And I think certainly cardiovascular drugs, certainly neuroscience, neurodegenerative medicines where orals are preferred, given that they can cross the blood-brain barrier much more efficiently, are going to be areas that are under strain. And it won't happen overnight, but I think step by step, you're gonna see that. Certainly in our world, we're heavily focused on trying to improve the legislation in any way we can, because I think getting 9 to 13 would enable companies to have time. But if you look at our own experience, a drug like Entresto only starts to pay back in years 7, 8, and 9. So if you only have 9 years, it's very difficult in a major cardiovascular drug to get the payback. And even if you look at our own examples again of Kisqali, the adjuvant study, if you had started that today, it'd be very difficult to do Kisqali in early breast cancer and not run out of the clock. And so I think this is going to have a significant impact. Unfortunately, we'll only see it 5, 6 years from now, and then it will take a decade to correct. And that's what policymakers really have to understand.
So a couple more questions. You and I have talked several times over the years about radioligand therapies. I will say Novartis was really one of the early movers there when, others were not there. That field has now blown up, but you guys still have to stay ahead. Or, I assume that's a goal of yours. So there are various things that are going on in that field in terms of innovation, radioligand, manufacturing, and where do you see your sort of main emphasis on maintaining leadership in that field, or at least maintaining your position in that field?
So first, in market, we continue to see significant growth, both from Lutathera and Pluvicto, which continue to expand to more and more indications. Lutathera now moving to the frontline setting in neuroendocrine tumors. Pluvicto having a soon to be filed in the earlier metastatic setting, and then moving into earlier and earlier lines. There, we're really going global. We'll have 3 manufacturing sites in the US, 3 sites in Europe, and sites in Korea, Japan, and China. So we have a significant global manufacturing footprint. So we really want to make sure that global footprint is there to deliver the therapy. Then our biggest focus right now is lutetium-based new targets. And so we have a range of development programs, primarily lutetium-based, that are going after novel targets, trying to get after other cancers, but also life cycle management as well. Lutathera and small cell lung cancer is one example. We take a FAPI, a newer agent that we in-license, and we take that into a range of solid tumors. And currently in basket studies to really see where the biggest opportunity is. So really cover the target space with lutetium-based therapies. And then we also now invest in actinium-based therapies as well. And I think as we've discussed in the past, beta emitters give you a much more broad coverage of the tissue, but less focused, less powerful energy near the molecule, but give you more reach. Actinium is or alpha emitters are much more targeted, but you get less reach into the stroma of the tumor. So there, with the recent acquisition of Mariana Oncology, we get an actinium-based profile pipeline. We already have, in fact, 2 pipeline PSMA. We don't get much coverage for it because everyone's focused on Pluvicto. But we're already in phase 3 with an actinium-based PSMA follow on to Pluvicto, and another one in phase 1/2. So we're already making sure that in the PSMA space, we cover actinium as well. And we think with that, with an alpha and beta emitter, we'd be in a pretty good position.
And your manufacturing strategy accounts for both of the different radioligands, because obviously that's an issue as well.
Yes. So for lutetium, we're very well covered, as you could imagine. We have the capability, but we need to, of course, scale up within actinium. But one of the things we've really invested in is having world-class scale in manufacturing. So we're semi-automated or fully automated. And I think some of the new competitors coming in will have to go up a learning curve in being able to deliver these doses, as we did. But now we're at a scale where we can make hundreds of thousands of these doses and deliver them around the globe. And you have to do it within 5 days. And we're able to do that consistently. In the US right now, we're over 99.5% on time in full delivery to the patient.
So last question, I can't let you get away without holding you to account for your goal to become one of the top 5 or 7 pharmas in terms of US sales. You knew this was going to happen. We had marked Scemblix, in fact, as one of your best opportunities, but I don't know if you see it that way. How do you see the 2 programs we started with discussing in terms of your goal, and how are you tracking to that?
We're tracking well. I mean, we had one of the highest growth rates in the industry last year in the US. So we grew in the mid-teens. So I think that puts us in a very good trajectory. Clearly for us now, it's going to be a combination of the inline brands, particularly Leqvio, Pluvicto, and Kisqali, that really have to fly. And then our upcoming launches, as you mentioned, Scemblix, iptacopan, and Fabhalta, which is, and Remibrutinib. Fabhalta and iptacopan are the same drug. Those 3 in terms of launches. And I expect in the 5-year period, we should get in the range of 5 to 7. Will we get exactly to 5? A lot of it will depend on our competitors and how their LOE cliffs go. But I feel confident that given that we had 10 positive phase 3s last year, given that we are growing double digit in 2023 and through first quarter of this year, still growing double digit, we have globally and in the US, we are on the trajectory with our focus strategy to get there. And I think we can do it. And I'll look forward to continuing to discuss it with you in the years to come.
We will certainly do that. Well, this has really been a great conversation. Thank you very much. And we look forward to another time. Thank you.
Thank you, Simone.